Okay, should we get started? Okay. All right. Hi, everyone. And here comes our fifth speaker. Great. We're all here. Well, I want to welcome everybody. I'm Marcy Darnovsky from the Center for Genetics and Society. And this is GMOs 2.0, Reengineering Life from Plants to People. This webinar is actually a collaborative project of all the people that you see on the screen and organizationally of the Center for Genetics and Society and Friends of the Earth. And our goal in this webinar today is to begin to familiarize all of you with the social implications of a new generation of genetic engineering techniques that has the capacity to redesign plants and animals and ecosystems and human beings. We have today 4 wonderful speakers, and each of them will comment briefly about a different aspect of this next generation genetic engineering suite of tools. And after that, we'll have a good chunk of time for your questions and your comments. Now, all the participants' our lines are muted so that we can maintain good audio quality for everyone. We have a big crowd with us today. But at any time during the presentation, you can submit your questions and comments, and the way to do that is to type them into the Q&A box that you see at the bottom right of your screen. And I want to let you know that we're recording this webinar so that you can view it again and that you can share it with friends and colleagues. And after the webinar, we'll be emailing all of you and sending you a link to the posted version, which will be up in the next day or two, and also links to more information from each of the organizations and speakers here. So we'll start with some very brief introductions. I'll introduce each of the speakers, and then we will turn it over to them. So our first speaker is going to be Doudna Perls. And Doudna is a food and technology campaigner with Friends of the Earth. And she's the co-author of the recent joint report from Friends of the Earth and the Center for Genetics and Society. That report is titled Extreme Genetic Engineering and the Human Future, Reclaiming Emerging Biotechnologies for the Common Good. And Doudna's going to start us off with a broad definition of what we mean by synthetic biology and gene editing, what they're being used for, and why they're important for environmentalists and social change advocates to be aware of and keep track of. The next speaker after that will be Ricarda Steinbrecher. And Ricarda is a molecular geneticist and developmental biologist. She's currently the co-director of EcoNexus, which is a UK-based public interest and scientific research organization. And she has been working since 1995 on genetically modified organisms, on their risks and potential consequences on health, food security, agriculture, biodiversity, and ecosystems. Ricarda is going to give us a brief overview of the new techniques, how they're different from previous generations of genetic engineering methods, and with a focus on gene editing, and especially what you may have heard of, CRISPR-Cas9. And she'll also be touching on gene drives and say a few words about the claims to precision that are made to these new tools. After Ricarda, we'll hear from Jaydee Hanson, who is the policy director at the Center for Technology Assessment, where he focuses on human genetics, stem cell research, cloning, gene and embryo patenting, and the convergence of biotechnology and nanotechnology. Jaydee previously served as staff director of genetics and bioethics issues and as legislative director for the United Methodist Church. And Jaydee's focus today will be on a couple of specific applications of gene editing and gene drives, and he's going to talk to us about ongoing and planned experiments with mice and mosquitoes, and he'll make some broad recommendations for public policy. Our final speaker, before we bring in questions and comments from all of you, last but not least, is Shanks, who has worked at the Center for Genetics and Society since its beginning, which is in the early 2000s, in a consultant capacity. Pete is a regular contributor to the Center's blog, Biopolitical Times, and he is the lead author of the Friends of the Earth Center for Genetics and Society report that I mentioned before, Green Genetic Engineering and the Human Future. And Pete will turn our attention from genetically modified plants and animals to the prospect of genetically modified human beings and tell us about the controversy that is currently raging, I think it's fair to say, about the prospect of genetically modified humans. So again, for those of you who just joined us, since I began the introductions, I want to point to the question and answer box that's at the bottom right of your screen and invite you to type in their questions and comments at any point during the presentations, and we'll turn to them after all the presenters are done with their brief comments. So with that, I'm going to turn it over to our speakers, with Doudna first.
Thanks so much, Marcy. We are at a societal crossroads. It's a crossroads for our food system, for ecosystems, and for what it means to be human. We have the opportunity now to choose the path of responsible technology and a healthy, democratic agricultural system, balanced ecosystems of animals and insects, and appropriate and careful uses of technologies within human beings. The current technology platform proposes risk further entrenching corporate control over agriculture, nature, and humans, and really might entrench us into industrial systems that we know are not working. Many of us have learned about GMOs via agriculture. Although GMOs have been touted as safe, sustainable, and a way to feed a growing population, evidence really demonstrates otherwise. 90% of GMO crops in the US are engineered to be herbicide tolerant, and we've increased herbicides use by hundreds of millions of pounds since 1996. Unfortunately, there have been many unintended consequences as a result because we have rushed into widespread use of these technologies before broad-based public discussions, robust analyses of direct and indirect impacts, or oversight, regulations, and transparency. And now we're hearing the same stories that we've already been told with this next generation of genetic engineering, also known as synthetic biology, a set of new technologies that are rapidly entering the marketplace, driven and patented by biotech companies. This spectrum of applications is essential in understanding of what it is we're facing moving forward and looking at extreme applications such as GMO humans. If we're going to care about GMO corn, for example, we need to pay attention to and care about GMO insects, animals, and even GMO humans. So what is synthetic biology? How does it work? Instead of swapping genes from one species to another, as in previous genetic engineering, synthetic biologists may draw from a variety of approaches. These can include creating new genetic materials such as DNA in order to create or reprogram new life forms. For example, in one current commercialized application, scientists might write entirely new genetic code on a computer, print it out, and then insert it into yeast organisms. This synthetic biology yeast then feeds on sugar and biomass to produce an artificial vanilla flavoring, or stevia, for example. This science is very complex, and yet it's already entering the environment and market. There are no safety assessments specific to these new techniques and no regulatory oversight for a very swiftly moving set of technologies that no one knows how to predict or control. Synthetic biologists have their sights now set on plants, insects, animals, or microbes that could produce fuels, industrial chemicals, medicine, and ag crops. This set of genetic engineering is more risky than the first generation GMOs. There's a greater detail of intervention, faster speed of creation, novel organisms and DNA, and many more unknown consequences. We're really continuing to see also the corporate consolidation of companies which are seeking to create, patent, and profit from life. Synthetic biology giants like Intrexon owns the GMO apple, salmon, and mosquito companies, as well as animal cloning, DNA fabrication, and more. So what's next? There is a next generation of GMO crops and animals and insects designed to fit into the industrial agriculture system, or designed to eradicate plants or animals we consider to be pests or invasive. It is this new genetic engineering industry that could undermine regenerative organic farming, sustainable humane livestock production, conservation of ecosystems and biodiversity, many of the things we're working to promote. We are seeing these new techniques proposed for engineering humans as well. This past winter, as Marcy mentioned, we released a report on GMO humans with the Center of Genetics and Society and Friends of the Earth. It looks at health, regulatory, social, and ethical questions. What about regulations? This synthetic biology research is galloping ahead with virtually no oversight, regulation, or overview to anticipate unintended consequences. Industry is pushing for less regulation. We're seeing a debate around language. What is genetic engineering? We're seeing an attempt to squeeze new techniques into decades-old regulations that are inadequate to address the new technologies and the potential risks of engineering nature. Before these new techniques enter the environment, our bodies, or the market, we need public dialogue, regulations, transparency, safety studies, and honesty about the whole system of production and potential risks. It's important we know what this new generation of genetic engineering is so that we can advocate for responsible technology that adheres to the values of sustainability, safety, and social equity. And now I'll turn this over to Ricarda, who will talk about exactly what these techniques are and how they work.
Basically, I was looking at what has really changed in the meantime since we. Oh, there we are with the slide. Since we've last been looking intensely at the genetic engineering applications, and we can see that. Because the knowledge has increased, the technologies have been refined, new methodology has come to it. Like we have also like a higher degree of data throughput systems, robot systems. So altogether, it's now possible to have really to make much deeper and more complex changes in the genetic makeup and in the metabolic pathways of living organisms. It's like it's a very deep sort of change or like interference that is now possible to the extent that organisms can be completely reprogrammed of what they are used to be doing. Mostly that is with microorganisms currently, but of course the research is going on in different directions as well and towards other organisms. Now the other aspect is targeting special DNA sequences, that there is a higher degree of precision on that level, which means like this is the so-called gene editing tools with the so-called site-specific nucleases, and I'll be going more into detail there in a while. And the third big change is, and it has already been mentioned, the ability to now not just alter an individual organism, but to actually apply this across populations and even across ecosystems with the gene drive system. So we basically have entered now this area, this time of synthetic biology and also of the so-called new breeding techniques. I'm saying so-called new breeding techniques because it's got that word breeding in it, and it's actually got nothing to do with breeding whatsoever. It's a name given to it by those who have developed it, partially by industry who's really interested that it will not be recognized as genetic modified organisms. So we have to be careful when we use this word new breeding techniques or NBTs, and as you can see in my next slide, I prefer to call it the new genetic engineering techniques. With these ones, there's quite a breadth of them around, but we can categorize them maybe into three sections. One area is that of the genome editing techniques, those techniques that will actually target directly the genome, the genes, in a precise in an intendedly precise manner, meaning it's targeting specific DNA sequences. Then the second area is that which still applies the traditional or the current GE techniques, but it looks, for example, to have the sourced material to come from sort of closely related species, and that would be the cisgenics or intragenics. And there's other methods as well to, for example, make sure that after the process of genetic engineering, the product itself will no longer have a GM sequence in it. That has been around for a while, that idea, but it's been worked on further. The final area probably, and that is a big area of research, is to not alter the DNA itself, but the surrounding of the DNA, the wrapping, the outer side of the DNA, and thus change the gene regulation. It's part of a field called epigenetics. And this can have also very, very profound impacts, but I will not go into that today. Let's just quickly recall on a lot of you who will have worked on and looked at GMOs in the past will be familiar with this kind of description. Basically, if you look at the plant, and just to see like which level are we actually talking about, if you look at the plant, take a tissue like a leaf, then you can see the cells. If you look at the cells and look deeper into the cells, then you can, I don't know what just happened here. Then you can see the nucleus, and in the nucleus is all the DNA, which is all the inheritable material. It's the command center, basically the control center of the cell, and therefore it has all the information required, how to regulate and how to pass on information. And in that you have these long threads called the chromosomes, which are made up of the DNA, and on the DNA you have distinct areas which we refer to as genes, which are areas of distinct function, for example, of producing certain proteins. But if you look even closer down, if you really look at the very linear, very naked level of the DNA, you see it's made up of individual letters. There's 4 of them, and it's A, C, T, and G. These are the nucleotides, and they link together as pairs and making a very strong double strand, and that is then the double helix. It is at that level that we are talking about when we are talking about the new genetic engineering techniques. But I come to that in a moment, because let's just recall the previous versions of genetic modification, because some of that is still being used. Even the genetic gene editing components still need to be delivered into the cells with these transformation techniques. So transformation, what does that mean? We are needing to get the gene construct into the DNA, into the cell of the plant. And here you can see the example. If you put it into this ring of the agrobacterium and use that as a shuttle, or if you produce lots of it and put on small gold particles and you shoot them into the cell, it's the shotgun techniques, you will end up having your gene just, it could end up anywhere on the chromosomes, on the DNA. You can see here like the little gold points or the little white long squares. You don't know how many you have in there or where they have ended up, or if some of them might just have tried to enter and injured the DNA and just left again. Basically, the result of that is that we have random integration, we have imprecision, and we have hundreds to thousands of mutations. I just saw a note here, there is no audio. Is there an audio right now? Yeah? Okay. Okay, fine.
Ricarda, you're fine. Jane,
okay, fine. So basically, this technique has shown also how important it is. I mean, this research into the background mutations occurring here also shows how important it is to actually look at the process of genetic engineering and not just at a product level. And so moving on from here, basically like having all these unintended side effects, these extra injuries that need to be bred out, then all these extra levels of risk assessment to completely unexpected and unintended changes. What could be the answer, both from the level of making it more easy to predict, but also to maybe not have to have such a burden of risk assessment? That has been part of the ideas behind that research. Now, so what could have been done? So basically the idea was then to design something which could be described as molecular scissors, sort of these so-called nucleases. They cut the DNA, and if you can enable them to actually find specific DNAs, if you could make them target DNAs at a specific level, then you would have something that cut the DNA at the place you want, and then you could try to insert the gene you want at that place or alter it at that place. So that is the idea of precision behind there. I will come back to that in a while as well, because there is this notion of precision equals predictability. But on which level would that predictability be? And would that also mean like there is no unpredictable results, which is not the case, and therefore this notion precision equals predictability equals safety is a wishful thinking, but it is not backed up by any kind of scientific evidence, nor by the logic in itself. Now let's go to genome editing. As I said, you can see here, like, I want to target that particular piece of the chromosome where it says gene to target. What do? I will basically determine the sequence of the target DNA, and then I will have to design or choose a specific cutting enzyme that is good for that. Like the CRISPR-Cas system has, over the last 2 years, really been refined, that it can be used like all across the different organisms and species. And so that is then equipped with the target sequence as a sort of home-in device. And you can see here that basically like this bluish-greenish entity is that enzyme that will then, with the help of the target sequence that has been built into it, it will find the specific site of the chromosome of the DNA and do 2 cuts, like basically do a double-strand break in the DNA, and then you can either put new DNA in it, like the donor DNA, or you can sort of initiate mutations, or for human gene therapy, it might be you want to repair the DNA there, or you can cause deletions, insertions I've mentioned. And this step, actually the so-called repair step, or the putting the DNA back together again, is done with enzymes that the cell has itself. That is part of the repair, so-called repair mechanism that all organisms have within them. Yet these specific targeting cutting enzymes that can be sent to specific places need to be introduced, and they are commonly introduced on a piece of DNA, on a plasmid, and still have to be transported into the cell via agrobacterium or ballistics. And so usually they get also inserted into the DNA of the host organisms, and thus it is clearly, on that level alone, genetic engineering. And from there then, this can act and do the genome editing. Now, unpredictabilities of risks, off-target effects basically means that the DNA can be cut at places it was not intended. It can mean also that, which is the off-target, as I said, which can alter the function of the proteins, of different other proteins. It can also change the expression of genes if, for example, regulatory sequences have been injured. Then, of course, then you have the problem of unintended integration of the added DNA. But, of course, we also have all the risks of interactions of the genetic sequences introduced and how they will now impact of the whole metabolic pathways within the organisms. We have done a little report on that, and if you go to our website, you can have a look more at the details. Now, don't worry about this slide. I'm not going to explain it all here. This is just an example of, you can see a few distinct metabolic pathways. If you alter something in one would think, oh, that will stay in it, for example, in the bottom left blue one or in the green one. But you can see that all of them actually have an interaction with other pathways in there. So what that means is the idea that one can change something precise and things like, okay, I've changed that particular enzyme, I've changed that particular protein, that gene, there is no guarantee that it will only work in that function you know about. It can have so many other functions we do not have an understanding of. So therefore, the idea of precision in that is vastly exaggerated because we do not have the background knowledge. So the idea of precision, as I just said, is based on that one knows what one is doing. Here, this is not the case. And basically, we only know about the level of nucleotides of the sequences we put in. But what is missing is the whole contextualization into the genome, the epigenetic landscape, the organisms, ecosystems, or also the socioeconomic conditions. And this precision around nucleotides gives us really a false sense of predictability and safety, as I have mentioned before. So from that briefly, I move on to the gene drive, which is using these technologies of CRISPR-Cas. And what one can do with that is basically not just alter one organism, but make sure that the gene goes across to all other members which are sexually reproducing together. So population scale genetic engineering, ecosystem engineering. And where is that being applied? It's applied to crops and weeds, for example, weeds that have inherited or have developed now and pass it on like resistance to certain herbicides. In order to make them susceptible again, there is the idea to basically put a gene into them that will undo their resistance and let that spread across, so therefore they will be susceptible to the pesticides again that farmers will spray. The side effects of that are not being taken into consideration here, but also there is of course the idea of just weakening weeds or of weakening invasive species, making them susceptible to diseases, making them more prone to different stresses. The consequence of which of course is it will not just stop at where you want to have it, like at the farmer's field, for example, but invasive species also are home somewhere. Weeds are plants that also are members of an ecosystem elsewhere, a full integrated member. So if that gets weakened, we actually really create big problems. Mostly, and I think Jaydee will talk about that, it's currently being discussed about with regard of mosquitoes. So my following slides will be about mosquitoes, but it's a big intention to use it against pests, invasive species, weeds, etc. So just to explain what it is and how it works, usually in reproduction there is something which is like the Mendelian Inheritance, like about 50% will pass on a trait, like here in this case with the blue mosquito will pass on if it combines with in this case a gray one, the reproduction will lead to a 50-50 of blue and gray. But you can see that it will, if there is more gray around, it will drown out the blue in this case. Now, with a gene drive, you will make sure that when the gene which comes from the blue comes to the parent chromosome of the black, or of the gray one here, it will actually push its own gene across. I'll show that in a moment later. And what happens is that by this, every single one of the offspring will have the inherited trait. And will have the trait I originally put in, the trait I want to drive through all the populations. So in this case, just to show you how it works, you use this, for example, the CRISPR-Cas system. On the left, you can see you have that blue part that also weakens a particular gene, which is here sort of in red. So if you then have the gene drive system built into that, it will cut the DNA of the other chromosome at that place where that gene sits for the trait and put itself across, and will do so in every generation and across all the cells. So that is behind the gene drive system. It had been a dream for a long time, but time up.
Ricarda. Hi, Ricarda. This is Marcy. I just want to draw your attention to the time. So if you could wrap
Thank
up
you very much.
slide,
so basically, this is now no longer something that is sort of an idea. It actually has distinct research successes behind it, and therefore, because of all the consequences in that, we need to pay attention to that. Last slide. The concerns and risks, and I think Jaydee will go into that in a moment as well, is of course the impacts on biodiversity if that spreads into ecosystems. But also, like, if I take just members of an ecosystem out, the impacts on humans, the environment, food security, because it can be used to crops as well. And also we should never forget, all these things can also go into so-called wrong hands. And there is a grave concern in the community around bioweapons, because especially CRISPR-Cas, the gene editing techniques, plus combined with gene drive, is a very serious concern. That's it. Thanks.
Well, I will move.
Okay, thank you so much.
Yes, I will move quickly through the slides because I realize we're running late. A lot of the researchers planning to use these CRISPR and gene editing techniques want to edit the genomes of wild animals. This is very different than even editing the human genome, where we're working on just one family and not anticipating it to go throughout the entire ecosystem. Unless these new ways to edit genomes are well controlled, we could seriously damage the ecosystems. Scientists have already used CRISPR to edit fruit flies. They're moving on to other organisms rapidly. They've already been using it to edit actually one breed of cattle, but we're not planning to release those cattle into the whole population. But when you move to something like a mosquito, and you're introducing traits in a way that bypass normal inheritance, the genetic changes take place very quickly. One of the proposals is to edit the Anopheles mosquitoes that transmit human malaria. And the proponents argue this only makes a small change to the ecosystem, but these insects have particularly in the tropical world, have a big part in the ecosystem. That doesn't mean that I'm a big fan of malaria. It means that we need to look at disease as part of a social system. As we evaluate these techniques, we need to make sure that we don't allow the release of applications that can go wild, pun intended, on their own. And one of the applications that's being considered is to engineer white-footed mice because they're one of the rodents that carry the Lyme disease. The problem with this is if you do it even on a little island off of Massachusetts, these mice can hop aboard ships, and pretty soon you're editing all the mice in North America. And what's worse, they're not the only rodent that carries the Lyme disease. So are we going to do chipmunks next? All other mice next? It's a huge problem, and the public has the right to know and participate about whether these techniques should be used. It's interesting that the company Oxitec, which has a GE mosquito not developed through these techniques, has mostly been releasing its mosquito in countries with fairly poor populations and weak oversight, Brazil being one of the cases. The FDA used to require a hearing, a public hearing, before any GE insects and animals were released. Several of us went to meet with the FDA to remind them of this requirement, and they deleted that requirement from their policy. So the genetically engineered mosquito to be released in Florida won't have an FDA hearing. The U.S. Department of Agriculture neglected to give public notice that it had approved the first release of GE moss, but we're going back and challenging that failure of the law. We believe that any company or university that plans to release wild animals or insects should be required to obtain liability insurance specifically to the release. It will be interesting to see to what extent they're able to get such insurance. The experiments involving gene-drive organisms could keep harming the environment for years after the release, whether intentional or accidental. Think gypsy moths, which were an accidental release, but are still moving through oak forest in the eastern United States, or kudzu, which was an intentional release and has moved north, and with climate change is moving still further north. We think the developer should pay for all of these cleanups. That there shouldn't be blanket exemptions from good environmental review, because the ecological and economic risk of each application are unique. Cane toads, an invasive species throughout the world, pose different risks than GE mammals, which pose different risk from GE mosquitoes. We are very suspicious about the containment that some of these proponents are proposing. Biological containment won't work. Even with the GE mosquitoes that are being released by the company Oxitec, they're only getting 90% sterilization. Physical containment won't work either. One idea is that you would just test the mice on a remote island, but the mice, as I mentioned before, easily hop onto boats, and we already have a problem that wild-type mice get into the labs of pharmaceutical companies that are breeding special lines of GE mice, and the wild-type mice get in and breed with those mice. My assumption is that the other GE mice would do likewise. I will skip this. We need to take a very precautionary approach. If there's another way to do these kind of things, it should be done. Mosquitoes carry viruses and malaria, but most deaths from these diseases are poor people in countries ravaged by war. GE mosquitoes did not eliminate malaria from El Salvador in 20 years, but good public health policy did. My friend Eleanor Powles at the Wilson Center admits that we now have the power to hijack evolution. People think this will be predictable and efficient. It's not the case here. We need to understand how to talk to the public so they understand these risks. Currently, one of the poster children for why you should do genetically engineered mosquitoes is Zika. And it's interesting that the province of Brazil, where they've had more problems with Zika, is a place that has extremely limited women's reproductive health services. And wrapping up, finally, I think it's important that the public recognizes hype. Overhyping these techniques by their developers will lead to greater suspicion. Even the developers of these CRISPR and gene drive techniques recognize this. I would say the FDA doesn't, but the developers do. MIT's scientist Kevin Esvalt, who's working on engineering mosquitoes and mice, lifts up the challenge of Zika. If Zika is what it takes to talk seriously about gene drives in advance of the experiments so that public conversation can demand that those experiments are transparent, that's a silver lining to Zika, and we'd be fools not to grasp it and not to insist on, my addition is, and not to insist on serious public engagement and full transparency of all these experiments. Thank you.
Okay, Pete, please take it away, and then we'll just have a few minutes for people's questions. You can please continue putting your questions.
Hi, can you hear me? Right. I understand that this PowerPoint and maybe the other PowerPoints will be on the web, so I am going to skip through stuff quite drastically. If you take what the two previous speakers have said and then apply it to people, that's really scary to me. And hello, I'm not getting to the next slide. It's just slow. I want to make a distinction that's really important between germline and somatic. This is usually used about people, but in fact the discussion about mosquitoes assumes that it's a germline intervention, which means that when you make changes, the next generation and all subsequent generations have those changes. Somatic modification is what's usually called, in humans, gene therapy, which is a matter of making genetic changes that hopefully will revert an existing person back to a healthy state. And that's been way slower than expected. People were talking about it 25 years ago, but it's starting to come along. Now, I am getting a pretty slow response here. Human experiments are being done. In 2 publications out of China, and in the UK, a permission has been granted to do further experiments on human embryos. The UK one is specifically about developmental biology and seems to be reasonable. I am failing to move to the next slide. I don't know if there's a problem with this keyboard. Aha! I want to keep going. Maybe I just have to push hard. Sorry, this is really just to stress the urgency of having this discussion. This is not in the far future. This is happening right now, and there are literally hundreds of millions of dollars of venture capital going into the use of CRISPR gene editing, so theoretically, and in fact really, for medical purposes, but obviously doing that is liable to raise the risk of doing germline intervention. And the other distinction that we need to get in, I really don't know why I can't forward this slide, between therapy and enhancement. Therapy is basically curing a disease. Enhancement is what people talk about when they fantasize, and frankly at this stage it is fantasy, about producing future people with a low risk of Alzheimer's, with 20/20 vision permanently and such like. That's the picture. We move on. What Ricarda referred to this, we really don't know enough. Even on a simple thing like height, there are at least 697 genes, and they only explain 1/5 of the heritability of it. So if you change some of those genes, height might change. The other thing is, so will probably a bunch of other things, but we don't know what they are. The social concern, of course, is that someone will try and do it. There are, we've already seen with stem cells. Okay, let's go on to the next slide, because apparently someone else can do it. This is the no free lunch, which Eric Lander put up at some point, pointing out that certain genes lower your risk of one item and increase the risk of another effect. So you don't want HIV, but you don't want West Nile either. And distinguishing between those is, at this point, it's beyond the level of our scientific understanding. Next. See if we can get the next. I'm actually not going to spend as much time as I would have done on this. This is a list that's on the Center for Genetics and Society website with a great deal more detail about why each of these things is a serious problem. That if you're doing an experiment on an embryo and you don't know what's going to happen, obviously there is a profound health risk to future children. In most cases, in almost all cases, if people think they are likely to have a child with a particular heritable genetic disorder, actually selection of the embryo can get around that in almost every case. The next couple are sort of philosophical. People are not things. We shouldn't treat people as things, say I. And UNESCO and the European Commission, years ago, put forward this idea, which many governments have actually signed on to, that the human genome, taken on board, in a broad sense, is a common heritage of humanity, and no individual member of humanity has the right to adjust it, to change it. I just want to mention that we need science. We desperately need science. We need science to deal with climate change issues. We need science to deal with population issues and so on and so forth. we don't want people who are very suspicious about changing human genomes, so that all the public opinion polls show that, to get even less trustful of responsible science. Even if it worked, it will be for the rich. That's really what it comes down to. It would reinforce a stratification in society that I don't think we really want. Throw me onto the next slide and I'll wrap up. Oh, there's another one. This central question. Okay, we need governance, regulation, and control. A number of scientists have actively called for public discussion, public debate, and have called for not proceeding without social consensus. I'm right behind that. Some of them may be thinking they can persuade the public through debate to make an interventionist move. I don't agree with that. Okay. But the debate is absolutely necessary. And let's move on. NIH has said they won't fund it. Okay? Otherwise, this basically says what I just said. As you can tell, I'm improvising. Next, the National Academies are in a process. It sort of began publicly last December with a major meeting in Washington, D.C., which was attended by actually several hundred very distinguished, mostly scientists, also some sociologists, some ethicists, some, not as wide a group as we would like to see there. A lot of people were a lot of stakeholders, so to speak, were underrepresented. But they're continuing to gather public input through a combination of meetings and written applications. And I urge people to think about these topics, to go to the National Academies website where they will show you how you can comment. And I know what I'd like you to say, but it's much more important, I think, that you say what you want to say, and so that we actually can try and build to some kind of public consensus. The next slide shows the report that Dana and I worked on and released last December, which is available at the Friends of the Earth website and at the Genetics and Society website, or of course you could just Google it. It's a PDF. It's got a lot more in it, plus a lot of references at the end where you can pursue the information gathering. And the last slide I've got is, I think, a really kind of important quote from Jennifer Doudna, who's one of the discoverers, or in patent terms, inventors of the CRISPR-Cas gene editing system, who was also, to her great credit, part of the impetus to get the National Academies going to consider these questions, not just scientifically, not just medically, but also socially and legally. Because, as she says, once the discovery is made, it's out there. Anybody with basic molecular training can use it, CRISPR, for genome editing. That is a bit scary, which is the sentiment with which I heartily agree. Thank you.
Okay, well, thank you, Pete, and thank you all of our speakers. As you can hear, we had a very strong response to this webinar, a lot of participants. And I'm sorry that we had so much to say to you that we're not going to have time for questions. But here's what we will do. We will be sending all of you links to the material that the speakers talked about, and also we'll post their PowerPoints and written versions of their presentations online. And we will try to re-record the initial remarks by Dana so that those come through more clearly. So that will be available to you in the emails that we'll send around. And I'll just take about 1 or 2 minutes now to share with you some of the questions and comments that have come in from participants. So one of them follows right on what Pete was saying. The comment is, make no mistake, genetic engineering is a euphemism for eugenics. We also had questions about what counts as safe and how do we determine that, and that's of course going to be subject to a lot of controversy in each of the fields that these speakers talked about. Someone asked a very specific question about whether these kind of new gene editing tools make alterations that can be detected and what that will mean for regulatory purposes. So that's a very good question, and we'll post an answer to that question online. And someone else asked, why don't we use a precautionary principle paradigm instead of a risk management paradigm, which actually I think is more of a comment than a question, and a very good one. So again, I think we don't want to hold you too long, and we want to just thank all of you for joining us, and really thank all the speakers for sharing the wealth of information and for all the work that they're doing, and to the participants for all the work that you're doing. And